While I'm waiting for this, I'll say that this work is a part of a larger, can everyone hear me? Good, yay, lots of thumbs up, I'd like to see that. This work is a part of a larger collaboration and is the combined efforts of a lot of excellent scientists, including my former postdoc, Clara Quinn, my former graduate student, Robin Bagley, collaborators, Frida Lindstedt and Sarah Callum, and a lot of really fantastic undergraduates that have worked in my lab over the years. So warning coloration is perhaps one of the most widespread anti-predator strategies that we see in nature. And the basic idea is pretty simple. So for example, you have this brightly colored monarch butterfly that's toxic. You have this naive blue jay eating this monarch, and it leads to this memorably unpleasant experience for the blue jay in which he will not attack similarly colored insects in the future. And so obviously this didn't mean so much for this individual monarch, but the idea is if you've got a common enough signal, then the birds will learn to avoid it and the individual predation risk will be low. However, if you have a different morph coming in, then those should get hammered by predators. So in other words, there should be strong stabilizing selection on warning coloration. Despite this, we actually see a lot of variation in warning coloration within and between taxa of a lot of different animals, within and between populations of a lot of different taxa, which is pretty puzzling. And to understand this variation, we need to know something about the genetic basis of color, the demographic history of that color variation, and the ecological function of color as well. And so one potentially powerful system for integrating these approaches are pine sawflies in the genus Neodybryon. So these are pine-feeding hymenopterans, and as they munch on their pines, they sequester resins from their host, making them distasteful to a lot of predators. Despite the fact that they're all pretty similarly chemically defended, we have a whole bunch of variation in warning coloration from these highly cryptic morphs to something that's more aposematic and kind of everything in between. And so this variation combined with experimental tractability and a growing list of genomic resources make this a really excellent system for understanding warning color evolution. But that's a whole lot of variation to work with. So to start, we focused on variation that exists within a single brightly colored species, the red-headed pine sawfly. And so this is about 18 years worth of collecting data that started during my PhD and has continued into my lab. All these yellow dots mean that we have populations that look something like this, where you've got this brightly colored yellow larva with a bunch of black spots. However, in this neck of the woods and other regions of the mid-Atlantic US, you encounter this white morph that's a lot more common. And we've done some range-wide demographic analyses, so we know that this white morph is derived. Well, as a first step to understanding this variation, we need to know what the pigment is, because there's actually a lot of pigments that can make yellow. And so to do that, we did some simple biochemical assays, which told us that the pigment that we're dealing with is a carotenoid. And carotenoids are things that, like most insects, Neodybryon can't synthesize, so they have to obtain it from their diet. Once they get it from their diet, they then allocate it to various functions, including that bright yellow body color, but also things like visual pigments and immune and antioxidant defense. So we know it's a carotenoid, but we next wanted to know, is this a genetic-based difference in carotenoid processing? So to do that, we go out and we collect sawflies, we bring them into the lab and rear them under uniform conditions and on the same host. And when we do that, you can see that those brightly colored individuals from Michigan and white-bodied individuals from Virginia retain their color. You can see the quantitative data here. Going a step further, we can then cross these, and when we do that, we get an individual that's intermediate in color. And then through the magic of haplodiploidy, we can take that F1 female, take virgin females and have her lay haploid male offspring. When we do that, we get all sorts of recombination between the grandparental genomes and a range of variation in the haploid male progeny. So together, what these data tell us is that although this is a dietary pigment, the difference in color very much has a strong genetic basis. So going a step further, we can then take these haploid male progeny, we can genotype them, and ask what portions of the genome are associated with this color. So here are those QTL mapping results. It turns out QTL mapping in haploid individuals works really well. And so we have a couple of small effect QTL, but two whopping QTL that account for almost 80% of the variation in color. So going a step further, we can then say, all right, well, what might be under these regions that might be contributing to color? So we know that to get that fully bright yellow phenotype, what has to happen is the carotenoids have to get from the gut to the hemolymph, and then from the hemolymph to the target tissue. And so if we look under this first big effect QTL, we find this gene cameo2, which we know is involved in transporting carotenoids from the hemolymph to the target tissue. And this QTL is associated with this kind of muted yellow color, where you're kind of seeing the yellow in the hemolymph, but not quite permeating that outer layer. If we look in our second QTL, we see this lipid transfer protein that's necessary for getting the carotenoids from the gut to the hemolymph. And this is associated with a much brighter white phenotype, where we're not really seeing any yellow at all. And then as you might expect, there is a significant interaction between these QTL, such that if you already have this one, adding on this doesn't do much else. So obviously this is all correlational. We need to do some functional testing, but we think these are really good candidate genes. But of course, knowing the genes is only part of the story, right? Because then we have to explain, how do these white alleles spread in the population? In particular, how did a novel white individual overcome that strong initial disadvantage to being a rare color morph? And we have a couple potential explanations we consider. One of which is maybe you had an isolated bottleneck population and drift pushed things along that way. Or alternatively, perhaps there were some sort of selective pressures that compensated for that initial disadvantage. So first, to consider the drift hypothesis, we looked at population variation, genetic variation, and the idea being that if these were drift, we might find evidence of historical isolation in the white-bodied populations. So we collected individuals throughout their range, we sequenced them with RAD sequencing, and asked what the population structure looked like. So when we do that, we get a structure plot that looks something like this. We did detect two major clusters, shown in white and gray, but these had nothing to do with color. In particular, the strong break that we're seeing here really corresponds to the Appalachian Mountains. And then within this eastern region, we see a mix of all color, and we didn't detect any additional substructure there. So what that tells us is that there's not really any indication of a major demographic event that isolated these white-bodied populations. Going a step further, we can ask, is there anywhere in the genome that is consistently associated with color in these larvae? And so what I'm showing you here is we simply lumped together the white-bodied individuals and the yellow-bodied individuals and computed FST at every SNP along the genome. For reference, here's our minor effect and major effect QTL, and the punchline is we have a single FST outlier that happens to be this major effect locus leading to this white phenotype. So together, these are consistent with the idea that these color alleles are being maintained by selection. All right, so our data don't seem to support a major demographic impact, so we next wanted to ask, what are some sort of selective pressures that might have compensated for this initially rare morph? And one question we had was, well, maybe these white-bodied morphs are more effective signals to would-be predators. And we tested that in a couple different ways. The first thing we did is we used avian visual models to ask, from a bird's perspective, would this white morph stand out more against the pine background than the yellow morph? And so what you're looking at here are contrast values for hue against a pine background and brightness of the white and yellow morphs. And I know this is sort of a weird axis, but basically anything over 5 means that those should be highly conspicuous to a bird under almost any conditions. And so the punchline here is that A, both morphs are highly conspicuous, and B, they don't differ in conspicuousness. Going a step further, we then asked how naive birds would react to these differently colored morphs. And so basically what this assay looks like is we offer a hungry bird a sawfly on 3 subsequent trials. If they're sufficiently grossed out and learn to avoid that cue, they will start to reduce their willingness to attack. And so that's exactly what you're seeing here for this white-bodied and yellow-bodied morph, where birds are kind of going yuck over subsequent trials and quickly learning to avoid these. By contrast, if we give them another sawfly that is also a pine sawfly, also similarly chemically defended, but cryptic in coloration, the birds aren't learning their lesson here. So together what these tell us is both that color is important to the survival of these sawflies, and also that really there's no difference between the white and yellow morphs and their signaling efficacy. So then we decided to look at the source of the carotenoids and ask, well, maybe these white-bodied populations are on a host perhaps that are more limited in their carotenoid availability, and that might be another mechanism. And so we first had to ask, are they on different hosts in the first place? And so to do that, we went back to all of our historical collecting records and found that yes, indeed, white-bodied populations are especially common on pitch pine relative to other pines that are common in eastern North America. And so knowing this, we then asked, okay, well, does pitch pine have less carotenoids in its foliage? And the answer to that question was yes. So we found significantly lower carotenoid contents on this host on which the white-bodied populations are more common, which we assume to mean that the white-bodied populations have less access to carotenoids. So this led us to hypothesize that maybe trade-offs between the use of carotenoids in body coloration and these other important functions might have driven the fixation of these white-bodied alleles. Now, if that's the case, we might expect to see genetic correlations between white-body alleles and some of these other immune and defensive functions. So we looked for those. So we looked for genetic correlations between body color and defensive chemistry and quality and quantity, and also between encapsulation response, which is one of an insect's primary immune response against multicellular parasitoids. And so the punchline in here is we found nothing. There were no correlations whatsoever. However, we had fed, this is probably one of the very first things we did, and we had fed everything on a high carotenoid diet. So if this loss of yellow pigmentation really is adaptation to limiting carotenoid availability, those advantages might only manifest on a low carotenoid diet. So we're eager to follow that up. So we obviously have more work to do, but I think several themes are already emerging from this analysis. First, our genetic mapping data is consistent with this growing body of literature that color compared to other traits might be unusually simple and predictable, meaning that it's often due to a small number of large effect changes and a handful of genes that we see over and over again. Second, our demographic data tell us that it's not necessary to invoke a major demographic event such as a bottleneck or a founder effect to explain these shifts in morning coloration. And then finally, our ecological data indicate that there can be a multitude of selection pressures acting on color variation, and that for dietary-derived pigments such as carotenoids, the availability of those pigments might be a major source of variation in morning coloration within and between populations. And so moving beyond the single species, now that we know a little something about this, we can expand outwards and ask whether we see these same genetic and ecological and demographic themes when we look to these other species that share similar variation. And so with that, I'll thank again my collaborators on this work, my lab and funding sources, and a shameless plug. If you're interested in the genetics and demography and ecology of adaptation and speciation, come see me. I'm recruiting PhD students and postdocs. And if you want to hear more about our work, we have several talks coming up. So I'll thank you all for your attention. That's a great question. We have not done that yet. We would really like to manipulate the carotenoid content of their diet and do that, and just see what fitness looks like under different carotenoid conditions of these two morphs. But we would love to do that. This has always been kind of a side project in the lab and came together recently, but we would love to do that. Yeah. Yes?
Yeah. I'm wondering if the learning of the signal has to do with color. I think we've tried, or we're going to explore whether contrasts work. You could be one of the contrast area where then you can kind of see that color better.
That's a very good question. So all of these assays were done with kind of a printout fine background. So that's certainly something we could manipulate to see whether or not which cues are most important. I think also with these larvae, you've got the contrast within the larva. Well, I can talk more later, the black and the yellow, and I think that's important too. Thank you.